Hey guys, so I gotta start working on my schedule, get my schedule figured out better. But I'm meeting a few of you for picking up textbooks. Hi, Brandy, and they're available. We got actually another 10 or 15, so we're doing pretty good. If you don't have one, sorry that you had to buy one already. You got the syllabus. I'm not going to do too much of a discussion today other than just briefly go over the syllabus. And actually, at the moment, I'm waiting on my coffee. I'm going to go get that here in just a second, because if you know me, homeboy can't lecture without his coffee. That said, I gave you the syllabus. And the syllabus is kind of a mix of how things used to be and trying to recognize how things really are. And so I would say that the kinds of topics that are in the syllabus, that's what we're going to cover. And hopefully the schedule will be pretty good about what we're going to cover. We'll see. The exams, I'm still trying to figure out how I'm going to do that. My immediate thought is on exam day, I will drop an exam either by email or Moodle, and you will have 1 hour to open it and finish it and then submit it. And do it all at the same time, because otherwise you'll have somebody in your cohort that says, sure, I'll take the bump. I'll open the exam first. We can all look at it, and then you guys can open yours and do better, and then we'll swap. No, don't do that. So as we get closer to an exam time, we'll start talking about exactly how that goes. But that's how I'm thinking about doing it. I'm also going to start generating and putting up some study guide type things. As I said, I really want you to be thinking about your notes as a study guide, building yourself a study guide, going over the chapter and integrating what we're talking about into it, kind of the more important points. And let's see. So then we've got, at the moment, we have 4 tests, and we'll see how that goes. The first test is scheduled for, I think, September 18th, and we'll have time to review all that. So really, don't get overly concerned about that and the grades and things. Because we're online, I am not going to be a particularly evil grader. If you're keeping up and doing your best, we're going to be okay. One of the things that we do is at the on test days, part of the sort of extra points or ways of kind of making it so that you're not dependent on the exam is that you have articles, review articles, that we will talk a little bit about what kinds of articles would be appropriate, and you will write a small summary on the article. And it's a way of getting you to start looking at what's happening in literature about ideas in microbiology and disease, and picking an article that is appropriate to the topics that we're covering and seeing how that goes. I smell my coffee. I'll be right back. Okay, now I'm better. Oh, mother's milk. All right, so we've been talking a little bit about, one last thing. With the lab, I haven't figured out how we're going to do that yet exactly. I may set up some demonstrations and have you come by and look at them, or we'll post the pictures and things. On the off chance that we're still here and things start to settle down and the risk of, being in a closer space is less, maybe we'll meet up. I doubt that's going to happen. Sorry. But as far as lab goes, I'm leaving that kind of in the air for the moment. But we'll get you some stuff. And you'll see that in the syllabus is a rather large document. There is a fair amount of the usual boilerplate, attendance and participation, illness policy. If you're sick, don't cough on your computer screen, because who knows? No, I don't know whatever. Hopefully you're not going to get sick, okay? Take care of yourself. Let's see. What else is in here? And if you have nausea or vomiting, I don't know. You're going to be nurses. Figure it out. No. All right. So we're still talking a little bit of introduction, and we're going to be getting into some basic chemistry as well. Probably on Friday, I may actually indulge and do a bit more of an in-depth video tomorrow, getting into some of the basic chemistry and stuff too. But we were talking a little bit about some of the advances in disease epidemiology and microbiology in general, these different eras and these different ages. And we have covered the idea, of course, that until the discovery of the microscope and von Leeuwenhoek, the notion that there was little animalcules or small animals, small living creatures at the microscopic level was something that just wasn't known, and it was assumed that disease was transmitted by foul air or the vile humors or different things. And there's some interesting things that start to come about is that prior to the discovery of bacteria, and Pasteur's part of this, and von Leeuwenhoek's discovery of the microscope, there were traditions in which before a surgeon would open up a chest, they might splash spirits of wine, that would be distilled wine or alcohol, onto a patient's chest, thinking that that was the way I was taught, and potentially the shock of the cold lessens the shock of the initial incision. Of course, what they're doing is sterilizing the area. And so Dr. Lister, with the discovery that carboxylic acid, phenol as we know it, is a good disinfectant, comes about. Pasteur begins to do some really good experiments that are going to debunk the notion of spontaneous generation, which is another sort of way people thought things came about, that life begets life. That idea you can kind of imagine, where you have a piece of meat sitting out, and after a few days you begin to see maggots floating or crawling around on it. Well, we know what's happening is that the flies are laying eggs on it. The eggs hatch into larvae and they start crawling around. But the idea that it somehow has a life force that's able to actually spontaneously generate these maggots, I can see that. And it was only when Pasteur began his experiments of heating up a solution, a broth, in a glass vessel that had a neck on it, that he was sterilizing the sample, and the neck, even though it's open to the air, it's preventing bacteria and fungi and spores from falling into it, and it remains sterile. And he had to go through a couple of different rounds of experiments to make sure people recognize, or to debunk these, or to answer the criticisms of the destruction of life force. One of the initial experiments had a flask with what he called a wheatgrass or a straw infusion, which is basically taking farm straw, putting it into water, and it's going to have all kinds of bacteria in it. And he would boil it up, that sterilizes it, and then if you left it open, it of course would start to grow bacterial growth, and it would become cloudy. But if you kept it in this sealed container, which is now known as a Pasteur flask, it'd stay clear. And so what he initially had was a tube coming out that was then had a 90-degree angle on it. And he heated that tube to kill any bacteria coming in. And as long as nobody came in and blew on it, it's going to stay sterile. People said, well, okay, what you're doing is that heat, you're actually killing the life force somehow, killing the bacteria. And that's not, as good an experiment. So the next level of experiment was a Pasteur flask that now has an S tube that does that. And the bacteria and things that might be floating around, they actually accumulate down at the bottom. And as long as you sterilize the main container, it stays sterile. Apparently, at the Pasteur Institute in France, there are still Pasteur flasks that have the original infusion still, clear. I always figured it'd be amusing to go in there and just blow into it once, but they probably don't let you do that. Just a few more little thoughts that came in my mind. Just in my news feed today, I'm a huge space geek. A group of Japanese researchers have actually published an article demonstrating the fact that bacteria can remain functional and alive for, I think it was 6 years in the space environment. And that is a pretty amazing thing, and it's a testimony to just how tough bacteria actually are. These guys are pretty much badass. They can survive in so many different kinds of environments, and not necessarily the same strain of bacteria, but pretty much anywhere you go on the planet. Boiling water vents, the bottom of the ocean, at the top of Mount Everest, in the Arctic, you look for bacteria, you're going to find it. And, I had a student a few years ago who's now a dentist or in dental school, was interested in the bacteria in the mouth. And our initial experiment was just doing a census of the bacteria in the mouth. We rinsed our mouths, and we hadn't eaten anything particularly. We hadn't also brushed or washed with hydrogen peroxide or something. But the two of us would consistently have somewhere between 7 to 10 billion bacteria per mil of a 10 mil solution in our mouths at any one time. I mean, that's more people than there are on the planet. And that's just in your mouth. The number of bacteria that are covering you right now is probably somewhere in the trillions. There are more cells that are not you than there are cells of you sitting on you right now. Really, that's what you are, is just the carrying mechanism for all these bacteria that are riding around on you. They just have you wander around and spread themselves about. So we know the bacteria are able to live in space. One of the things that it looks like, the outer layer of bacteria on these sort of dried surfaces, they're taking the hit from like UV light, and they're not surviving. But the guys below, just a millimeter or so below, they are still kicking, and they are ready to be activated and cultured again. One of the things this means is that it's very possible that rocks and material from Mars may have made it to Earth, bringing bacteria. We know there are, in fact, rocks that are found in the Arctic, or in the poles, that are from Mars. And so this as a notion that there may be this spread of life. It's also interesting to note, we launched a few weeks ago the latest Mars rover called Perseverance. And the guys at the Jet Propulsion Lab, where they assemble the rovers, they take great care to sterilize these devices because they don't want to introduce Earth forms of life onto Mars. It's one of the things I think would be a bit ironic is the first time we find life on Mars, turns out it was a contamination that we brought there ourselves. So we'll be getting into some of these different ideas and things as we start talking about different kinds of metabolic pathways and things that the bacteria have. That's all for now. I promise I'm going to get better about scheduling myself up and organizing, but not today. It's actually something I think we all have to recognize, is that these are pretty strange times, and you got to give yourself a break. There's times where you go, look, I can't do everything, and at least between you and me, we're going to be okay. We're going to make this work, and hopefully you're going to get a pretty good understanding of basic biochemistry as it relates to bacteria and microbes and disease, and we'll get some good strategies for thinking about lab work as well. So that was 14 minutes, not much, but I probably will start getting into some biochemistry and give a bit more of an in-depth lecture between Thursday and on Friday, and have it for you on Friday. And we'll see about other things. There's a variety of different kinds of quiz ideas. I haven't started those out exactly. I'll start posting some stuff to Moodle, though, that will be a good resource for you. Keep an eye out for that. Until then, stay safe. Say hello to your bacteria. You start naming them all right? Good. And I'll talk with you later.